Our next speaker is Robert Liu from UC Berkeley, and he's going to tell us about telomeres, cancer, and the issue of how to get to grips with what's going on with the telomeres through sequencing.
Oops, sorry. I'll try to present somewhat quick. But anyway, I'm from the Hochmeyer lab at UC Berkeley, and as you might tell, I'm from Australia, and I recently started a postdoc in the Hochmeyer lab about 7 months ago. So I've been working in the telomere biology field for about over 10 years, and I'm going to present how I've started a project using telomere long-read sequencing to study a type of cancer that utilizes alternative lengthening of telomeres, or ALT. So as you can see on my introduction slide is there are different ways to visualize telomeres, and then you can see telomeres on metaphase spreads as shown by those red dots, or you can see telomeres on DNA fibers on this bottom image where the DNA is stained by blue and then telomeres are in green, and there's a overlapping EDU tract in red, and you can actually see telomere extension events. So I'll give an introduction as to what telomeres are. So because this is not a telomere conference, so telomeres are telomeres are repetitive sequences at the ends of linear chromosomes present in eukaryote cells. They, in mammals or in humans specifically, are composed of TTA GGG repeats, and about 5 to 15 kilobases in humans. One particular feature of the telomeres is that they terminate in a G overhang, which will become important later during my talk. And they're about 50 to 300 nucleotides long in humans. So telomeres, in normal somatic cells throughout your body, telomere length shortens over time with cell division. And this typically leads to one scenario where you have replicative senescence, which when telomeres are around 1 to 2 kilobases. However, in cancer cells, they can overcome this barrier by mutating tumor suppressor genes like P53 and PRB to continue dividing. However, this leads to a secondary bottleneck called telomere crisis, where telomeres become extremely short, about 100 or so base pairs. And most cells do not overcome this barrier and end up dying. However, stem cells, which I'm working with in the Hochmeyer lab, they have an unusual property in that they're repetitively immortal, and they are able to indefinitely be kept in culture and maintain. This is partially due to the fact that they're able to maintain the telomere length. How once you differentiate stem cells, they end up returning to a somatic cell lineage, and the telomeres continue to shorten over time, and then you have the same dilemma of you have potential replicative senescence. What I'm interested in is in cancer cells. The majority of cancer cells are able to reactivate a telomere maintenance mechanism, either the majority utilizing telomerase, which is a ribonuclear protein as shown below in this animation, which it reverse transcribes telomere repeats to the ends. What I'm interested in is the remaining 10 to 15% of cancers that utilize a homology-directed mechanism called alternative lengthening of telomeres, or ALT, where one telomere strand invades an adjacent telomere and utilizes it as a template for lengthening. So what particular characteristics of ALT is that they are defined by elevated telomere damage, where you visualize gamma H2X, gamma H2X first are shown in red, colocalizing with telomeres shown in green, and this is in metaphase spreads. You also get an increase of APBs defined by colocalization of PML with telomeres, and often they occur at large telomeres where the telomeres cluster together in APBs, and it is thought that APBs are sites of telomere recombination in ALT. Additionally, telomeres are very heterogeneous in ALT cancers where you have very long to very short telomeres. So this is visualized using a southern gel where you digest genomic DNA using restriction enzymes. But because telomeres lack these restriction enzyme sites, these restriction enzymes do not cut in the telomere, and then you can indirectly visualize telomere length. Additionally, ALT cells are in terms of the genetic background, is that a lot of alt cancers found clinically have loss of functional mutations in HRX or DAX. So HRX DAX is a histone chaperone protein that functions together to load H3.3 histone variants at heterochromatin. But additionally, HRX has affinity for G quadruplexes that has a particular importance at telomeres because telomeres are G-rich sequence repeats. And it's been shown that when you genetically ablate HRX, this increases the propensity of cells that are undergoing through telomere crisis to activate ALT, shown in this important paper that was published by one of my PhD co-supervisors. So in the Hochmeyer lab, there was previously published where they utilized a stem cell model which can inducibly activate ALT. So how this is done is that we have an HRX proficient stem cells which have you have HRX proficient stem cells as well as HRX knockouts. Well, we also have endogenous TERT removed and TERT then exogenously expressed on the LOXP site. So the reason why we have TERT on the LOXP site is then we can remove TERT at will in order to inducibly active ALT. And the reason is that you do not want to have telomerase in combination with ALT occurring at the same time, because this might complicate conclusions relating to telomere maintenance. So what you have these three scenarios where you have wild-type HRX, and then you differentiate them with the TERT in the conditional TERT express, and they remain telomerase positive. You can also then have wild-type HRX, and then loop out TERT, and then when the state, when differentiated, they remain mortal because they are unable to maintain telomeres in the absence of telomerase. However, because they're HRX wild-type, they do not activate ALT. However, our model is that with the HRX knocked out and then upon differentiation, that we found that they specifically induce ALT, and this is all shown on the next slide. So this is a TRF which I explained earlier, where you have wild-type HRX in the ES state, and then upon differentiation, they do not change the telomere length profile. However, when you have HRX knocked out, in the ES state, they are short. However, when you differentiate, they gain this heterogeneous and increased length phenotype that is phenotypically of ALT cells. We also performed APB assays, as well as looking for telomere synthesis inside APBs, which is another hallmark of ALT. And then both go up only specifically when you, in the HRX knockout cells upon differentiation from the ES state. So one of the outstanding questions in our project is to address how ALT telomeres lengthen. So to distort that telomeres lengthen via break-induced telomere synthesis mechanism, where a telomere double-strand break triggers strand invasion in another telomere, and then utilizes it as a template. But the key aspect is that you have conservative replication, where you have strand invasion followed by fielding synthesis of the invading telomere. So to answer this question, I've employed long-read sequencing, specifically nanopore sequencing. So nanopore sequencing is a technique where you have DNA that's adapted to sequencing adapter, and this allows it to localize to a motor protein, which then is threaded, and then you have a current that is read across the pore, and that current readout is then interpreted by a base calling algorithm, and this allows us to determine what bases are being pulled through the pore, and this base calling is essentially the principle of nanopore sequencing. However, we can also then employ other base calling of DNA modifications that I'll explain later on. So currently in the literature, there were two back-to-back papers that employed nanopore sequencing at telomeres, and they employed a teloseq method where they have a teloret that binds to the G overhang, but it binds to the terminus of the C strand at the bottom. And then they ligate a splint adapter followed by a sequencing adapter to that G overhang. However, due to the chemistry of nanopore sequencing, this only allows specific sequencing of the C strand. So this limitation then I've sought to overcome using a modified approach, which I will explain in the next slide. So in terms of how I'm going to measure telomere sequencing, measure telomere extension using nanopore sequencing, is to demonstrate conservative telomere synthesis versus semi-conservative DNA replication. So as I showed earlier, is that strand invasion is defined by conservative synthesis. So you have incorporation of nascent nucleotides on both strands. However, unlike semi-conservative DNA replication, you have incorporation of nascent nucleotides on either the nascent C or G strands. So in order to detect this telomere synthesis, we are going to be pulsing with BIDU and EDU, and then detect paired terminal BIDU-EDU tracks only at the ends of telomeres. So this is important because already as shown earlier is that telomere extension involves terminal synthesis. So this actually has then there's been work done in the field where the people have looked at DNA replication in general using nanopore sequencing. This is done by the Bomo lab. So they can actually pick up BIDU, distinguish it from EDU at replication origins shown there. And they call it DNA scent. So I'm going to apply this through combination with my method, which I call telobate enrichment. So this involves a telobate which is biotinylated on the 3' on the top strand, shown here, and then followed by T4 DNA ligation and T4 polymerase fill-in to fill in from the C strand of the telobate up to the junction for the 5' end of the telomere. And then followed, I perform ECOR1 restriction enzyme digestion and mobilization of strep beads, and this is then followed by hairpin ligation. So this hairpin ligation is important because this allows sequencing of both the C and G strand in order to demonstrate conservative synthesis and distinguish it from semi-conservative synthesis. So this is achieved by when you have the sequencing adapter that sequence on the bottom, you have the sequencing along the bottom, through the hairpin, and then back along the other side on the G strand. So I have preliminary data to show that this actually works. So I have a TRF where you have athenium bromide on the left half and then lack of athenium bromide on the right, and then you can see that telomeres stained by a radio-labeled probe are relatively enriched. And then this is in summary, I have some key cell lines, alt cells that are shown with those brackets, as well as the non-telomerase positive cells, the ES cells, as well as HRX knocked out ES cells that actually remain short, while my HRX knocked out fibroblasts, they actually gain I guess telomere smear, as you can see in the 6th lane. So when I correlate the eluate versus the import, you have some roughly correlation of the telomere length, suggesting that this enrichment technique at least works via TRF. When I look by qPCR, you have a similar trend where you have yields about from 10 to 30%, and a fold enrichment about 30 to 50-fold compared to genomic DNA. And the genomic DNA is amplified using primers against a multi-copy reference sequence MRF1. So this is actually some long-read sequencing preliminary results I've obtained across the variety of cell lines as you saw as well as other cell lines that you saw in the TRF. And this is for matched samples, so meaning that whatever samples that I've done on the TRF, I've used a portion of that and then used it for long-read sequencing. And you can see here that the similar trends are remain similar, where you have alt cells that I've labeled remain heterogeneous, but what you notice is that compared to alt cells that I've labeled compared to the other cells, like HeLa 1.3s, IMI90s, as well as the HRX wild-type ES cells and fibroblasts, there's a discrepancy between the mean and the median of those violin plots. So this is interesting, they're suggesting that alt telomeres have an abundance of short telomeres, leading to the skewing of telomere length profile that has not really been particularly pointed out in the field. So this heat map just shows that the telomere counts, the number of data points, so that is something I'm currently also working on. And you see that the correlation, though, compared to TRF, but eluate telomere length is not as good. However, that is something that is still being optimized. So this is finally just close to finishing off, is that this is some actual raw sequencing data I've obtained to demonstrate that the sequencing does work from the bottom strand, from the C strand, starting from the telobate, as highlighted here, and then this goes through the telomere C strand, then reaching the hairpins. So you're going through the subtelomere sequence, reaching the hairpin sequence, where you have in the middle, you have a composed of T repeats, because this is how the most efficient way to design a hairpin. And then going through the telomere G strand, and then ending in the telobate, top telobate sequence as indicated in the red box, and then the highlighted sequence. So yeah, in conclusion, I have some preliminary data that demonstrates that telomere long read sequencing is obtaining a high resolution telomere length of both short and long telomeres across both alt cells as well as our stem cell model. And we're currently optimizing read counts of telomere read counts as well as obtaining sufficient hairpin reads that are both telomeric and in the telobate sequence, as well as performing a BID and EDU base calling. And the future experiments involve lineage tracing in both our HRX null stem cell model as well as performing telomere proteomics. So finally, I would like to acknowledge my supervisor, Dirk Hockmeyer, as well as Jan, who has been instrumental in helping me start up in the lab, as well as former members, Tim and Antonio, who were co-first authors on the paper.
Thanks. I actually have a very general question here. Have you looked at the combination where the cells are telomerase positive and ATRX negative? Do they have super long telomeres, or which mechanism takes over, the ALT or the normal one?
Oh, yeah, you can have both TMMs operating at the same time.
Yeah.
So, but the problem is that any extension becomes hard to attribute whether it was due to telomerase or alt. So that's why we prefer to get rid of it to ensure that any extension must be due to alt.